Today's without wasting time because already 10 minutes over. So, in your syllabus, there are 3 topics. One is you describe the digestion and absorption of dietary proteins, and second one is hyperammonemia or ammonia formation, and third one is basically specialized products produced from the amino acids. So, I have decided I will take 7 classes. One will be on digestion of protein, which we will be discussing today. So, this class I will take one class on this one and 5 classes on this topic. So, today's topic will be basically digestion and absorption of dietary proteins. It can be covered in 10 minutes, it can be covered more than an hour. What I have told you in protein class, I think, do you remember? Have you read the protein from the books? Otherwise, you will not understand. See, proteins are basically polymers of amino acid joined by peptide linkage. So, that structure, primary structure is folded into a higher order of structure, secondary, quaternary, tertiary and then quaternary. Most of the proteins function in tertiary stage, and when the subunits are assembled, they are called quaternary stage. So, when we talk about the digestion of protein, basically we break down the protein into smaller fragments up to the level of its constituent amino acids. So, that is what is digestion. So, formation of protein was something where successive amino acids are joined together to form a polypeptide chain, and then it is folded. The reverse process, that is reverse process is the digestion. So, we have to break down. So, the first stage of protein digestion would be to break down that higher order of structures. If it is a quaternary structure, then quaternary structure. If it is a tertiary structure, that higher order of structures means the secondary, tertiary and quaternary structure are to be broken. Then the primary structure will be made available. So, the peptide bonds will be accessible and the peptide bonds will be cut. So, in the, if we, so if we remember that, that linear chain of polypeptide, so we are having one, see this is the structure of protein, okay. What is done basically, that structure is unfolded to a linear structure just like this one. This is how the polypeptide chain were formed. So, this is one is N terminal, one is C terminal. Okay. So, what is done? See, this protein needs to be fragmented. This protein needs to be denatured, otherwise these peptide bonds will not be accessible. We do not take protein in native form. Vegetable proteins we can take some Bengal grams or some peas in its as a raw material, but most of the animal proteins are cooked. During cooking, basically denaturation is done. Denaturation is done means higher order of structures are broken down. So, basically this is broken down and this is also fragmented. Into this type of structure. Okay. So, during mastication, when we chew food, these particular protein molecule are broken down. That food material, actually there is no exclusively food which is containing protein molecule until and unless it is synthesized. Even if we take meat, meat contains protein, meat contains also fat, also some amount of carbohydrate. So, there is no such food material which we commonly take. We take say egg also, egg is exclusively almost protein. One example I can give you, but still there is if you consider about the yolk part, so that is not absolutely protein. There are some fat materials are there. So, anyway, when we cook the material, protein peptide bonds are not accessible in the native state. That is why we require it to be denature, and denaturation is done mostly by cooking. It is by roasting, boiling, whatever way you say. Then that food we take and we then grind it with our, break it into small pieces so that the larger surface area are created. The digestion of any food will depend on the amount of surface area available to it so that the enzymes can act on those sites. So, more the fragmentation, more the surface area, more the accessibility of the enzyme, more the rapid digestion of that particular bond, and the amount of water how much you are taking along with the food also. So, there are many factors which affect the digestion, and because of all those things, the degree of boiling, the degree of denaturation, and the consistency of that food, particularly whether it is solid, semi-solid or a liquid, how you are taking, that determines the gastrointestinal transit time, means the time when you are taking the food in the mouth and it is basically passing through the intestine. So, we take the food, there is a temporary storage in stomach depending on the type of food what you take, and then from the stomach it is delivered into duodenum, jejunum, ileum. So, that is from stomach to cecum. So, that is called gastrocecal transit time. So, of course, something which is liquid, easily digestible will not be the same as a heavy meal which you take normally after a party. So, basically what I mean to say that protein needs to be denatured. So, at the end of denaturation, what you will get is smaller fragments. Even after this thing, you will get a structure like this. In case of protein, this native protein is basically, I am going a little fast so that there are many slides which I will show it to you. So, proteins is a large molecule. When it is broken down into smaller part, that is called proteose, and then still if it is lowered down, cut further down, it is called peptone, and then from peptone there is a polypeptide. Polypeptide is further broken down to oligopeptide, and then to several decapeptide, octapeptide, nonapeptide, whatever you say. At the end you will get a tripeptide, dipeptide, and finally amino acids. Okay. So, that is basically a protein digestion in short. So, protein will be broken down into smaller fragments. It will be called initial stage at the proteose stage and peptone stage, and then it will be further broken down to polypeptide, oligopeptide, shorter fragments of peptide, to finally tripeptide, dipeptide, and the amino acids also. So, the proteolytic enzymes, these enzymes which cleave the peptide bond are largely called two types of proteins, peptide bonds are there. One is protease, and another is peptidase. These are basically used almost synonymously. It is something like a large hammer and a small hammer. There is a big stone that needs to be broken down. That is, the big stone is a protein molecule, large protein molecule. Now, the hammer, which is of larger capacity, will break it down into smaller fragments. That the smaller hammer will break it down even to the grounded level, almost to the dust. That is what is peptidase, but they are called interchangeably they are used. So, there are bonds. First and foremost, this peptide polypeptide chain has to be broken into smaller fragments. How we can do? These internal bonds has to be broken at multiple points so that the polypeptide chain will be broken down. So, when the enzyme which breaks down the internal bonds are called endopeptidases. The bond which cleaves the internal bond of the peptide chain, this is endo. And at the end, there are two extreme terminal, one is N terminal and one is C terminal. So, as I told you, at the end you will get a dipeptide or a tripeptide. So, that this N terminal amino acid is cleaved from a dipeptide by an enzyme which is specifically named as aminopeptidase. Okay. So, this is an exopeptidase, extreme, that side, this is carboxypeptidase. So, aminopeptidases and carboxypeptidase are the exopeptidases. But all other enzymes mostly belong to the category of endopeptidases because they cleave the internal bond. So, like pepsin, trypsin, chymotrypsin, elastase, they break the internal bonds. They are peptidases, they are called proteases also. As I told you, if it is a large fragment, then it is called a protease in loosely used term, and a peptidase also. So, that is what is basically done. In the stomach, what happens? Food we take and the protein is mostly broken down into the form of proteose and peptone. And then it is propelled into the duodenum for further digestion, but basically polypeptide, dipeptides, oligopeptides, all types of things are formed, and that is further it moves, food moves further down the intestinal level, and the enzymes will act on that particular proteins. I mean those which could not be cleaved off or those which could not be broken down. The basic purpose at the end would be to get amino acids for final absorption. Only almost nearly more than 90% of proteins are absorbed by the portal venous, by the intestine. Only maximum 10%, around 10% protein may be lost in the stool. So, basically as you know that enzymes basically they require a pH, particular pH for optimal function. So, the stomach pH is acidic, it contains hydrochloric acid. So, that pH is conducive for the action of pepsin. Now, once the food reaches into the intestine, the pH changes to alkaline side. So, those enzymes which act on the duodenum, jejunum and ileum, they act on the alkaline pH, at the alkaline pH. They optimally act at that pH. So, body has an extensive mechanism how to regulate secretion or the enzymes from different organs on stimulation due to different foods. So, at the intestine what is happening? The amino acids are available, now they are absorbed. They are absorbed through the intestinal epithelial cells, mucosal cells. So, the mucosal cells when they reach into the intestine, there are mucosal cells. Okay, so now amino acids are available here. It is available here. So, amino acids are transported from this lumen, intestinal lumen, through the microvillus, which is present on the enterocytes, basically mucosal cells. They absorb it they transport it through an active transport mechanism or a carrier mediated transport mechanism. These are the two main mechanisms I will discuss in detail, and then it enters into the bloodstream and goes through the portal vein. Have you been taught about intestinal anatomy? Have you been told about intestinal anatomy, GI tract? Otherwise, you will not understand this thing. So, from the intestine, it will be absorbed by the portal vein, and then it will go directly to the liver. From the liver, it will be thrown into the main systemic circulation. So, once that anatomy part is clear, then if you read it, it will be easier to understand. So, hurriedly now I will go, this is the basic concept, this is the minimum amount of information you must have. So, basically just to summary, make a brief summary, that protein needs to be denatured, peptide bonds needs to be made accessible, to be cut by the endopeptides to make it into smaller fragments. The larger, the greater the fragment size, the greater the availability of the surface area, the greater the availability of enzyme action, so more substrate production, so less time for digestion, and there are many factors which affect digestion, how much, what is the boiling status, what is the denaturation status, how much you have masticated, how much water you have taken, because that requires a medium. Because if you take protein as dry, so that protein will not be soluble, so enzymes will not get a chance to act. Suppose if I take it as solid without breaking it, now how the enzymes will act? Enzyme will be able to try to find out peptide bonds here. Peptide bonds are not there. They are all almost inside. So, this needs to be broken down and it is to be made soluble, available, so that enzyme will act on the liquid medium. Substrates will be produced and the substrates will be then dislodged. Enzyme will act on that particular remaining part of the protein. So, what happens basically, the bonds are cut. So, they are made into smaller fragments and they are subjected to digestion by the endopeptidases. Now, whatever is not digested in the upper part will be digested at the lower part down the intestine. So, at the end we will get a smaller fragments finally to dipeptide, tripeptide, and there are specific enzymes available for breaking down the small peptides and to the amino acids, and these amino acids are absorbed. Okay. So, through the intestinal mucosa, through two main mechanisms, one is sodium dependent active transport, one is independent carrier mediated mechanisms, and third one is a Mr. cycle. So, one thing you have to remember here, one part, that what enzymes cleaves which bond, that is the crucial part, because enzymes are specific to cut a specific bond, but it is not that specific. As we say, enzymes are very, very specific. As you read higher up, further study, you will realize that all the enzymes are not that specific as we normally say. There is some amount of flexibility, and that does happen because in all cases, because otherwise so many amino acids are there. The enzymes which I will be discussing will cover only, it will not cover all, but ultimately the proteins will be broken down into smaller fragments finally to amino acid, that much you must remember. So, as I told you, native proteins are resistant to digestion because of the inaccessibility of the peptide bond. So, prior denaturation is required or by cooking, and the gastric H cell, that is an acidic medium, that also further denatures the protein and made it accessible to main proteolytic enzymes, as I told you, one is endopeptidase, one is exopeptidase. So, digestion occurs right from mouth to stomach, intestine, duodenum, jejunum and ileum, large intestine, but you see there is no digestion of protein in the mouth, because this mouth or the juice, that oral juice does not contain any proteolytic enzyme. So, protein digestion starts at the stomach and in the small intestine only. So, this food is passing through this stage as I have depicted. So, from this part to ultimately finally to the cecum, this is called cecum. So, that basically, and then the entire food will pass through this large intestine, which is colored as green. So, that is a total gastrointestinal transit time. Now, the gastrocecal transit time is from the basically from the stomach up to this cecum. So, that determines basically how long the foods are available in the intestine for digestion and absorption. So, in a living being, as I told you, the digestion, this higher order of structure needs to be broken. One is quaternary, tertiary and secondary structure. Then there will be sequential exposure of that protein to digestive enzymes. The digestive enzymes are basically hydrolytic enzymes because they hydrolyze the peptide bonds. So, peptide bonds will be hydrolyzed, means two fragments will be produced. So, there will be sequential exposure. So, cleaving of protein of large peptide to proteose and peptone, it is usually by the gastric juice, and then polypeptide, oligopeptide by the pancreatic juice, finally to tripeptide, dipeptide and to individual amino acid by the succus entericus, that is succus entericus means intestinal juice. So, then finally through the small intestine, the brass border villi, as I told you, this is not like that, the structure will be something like this. So, these border which I have written as straight line will not be like this. This will be something like dented structure or saw-shaped structure. So, villi structure is there. So, in histology we will know all these things. So, entry we take always food with some amount of water. So, total amount of water we take usually with the food is about 1200 ml and this is the breakup. So, total 8200 ml of water is absorbed. So, and the exit from the gastrointestinal tract, not the whole body, the food which is entering into the gastrointestinal tract. So, there are water 1200, this is saliva is there, stomach juice is there, pancreatic juice is there, bile, intestine. So, 8.2 liters of water is entering into the digestive system, and then through the portal vein almost 6700 ml of water is absorbed, and colonic reabsorption is there for 1400 ml, and with the stool we passes 100 ml. So, there is another balance of almost 100 ml. Now, how the urine is produced? Urine is produced out of this portal vein, which is the amount of water which is absorbed through the portal vein will be delivered into the systemic circulation, from where basically it will be filtered by the kidney and urine will be formed. Okay. So, pH of different juices are different because the enzymes act at different pHs. So, saliva usually is acidic, this saliva the pH is almost neutral, that is why we do not find any caustic taste in our mouth, either bitter something sour taste because acids are sour, but gastric juice occasionally perhaps you have seen that there is a degurgitation of food after taking a meal and you get some amount of taste of acid when the food, there is reflux from the stomach of food material into the mouth due to an abnormal food intake, or maybe during vomiting perhaps you have experienced there is slight sour taste of that content, that is basically the gastric juice is basically coming out during vomiting onto the oral cavity, but that to a great extent diluted one. So, pancreatic juice is alkaline and the succus entericus is the intestinal juice is basically, so that is the pH is 8.3. So, enzymes which are produced by the organs, they are produced in the form of zymogen. Zymogen, you know, they are the precursor protein basically. Actually, if the body does not have a mechanism of storing the enzymes in zymogen granules, the enzymes will be active within the cell, and there are intracellular proteins, including there are several cell membrane proteins also there. So, they will digest it. So, the cell will be digested. So, to prevent auto digestion of the cell and the intracellular breakdown of proteins, these proteolytic enzymes remain in the zymogen forms, while in the cell. Now, when they are released through a process of exocytosis, they are activated into the active form. What is that activated form? This zymogen granules, the active site is protected by a masked peptide chain. By masked, that particular active site is made masked by that sequence of polypeptide. That polypeptide or small fragment of oligopeptide is removed, as you have found during insulin. During my insulin class, I told you, insulin is secreted in the form of pre-proinsulin. Now, they are cut, and finally, the actual insulin, that is functional insulin, is produced. Similarly, here, there is a peptide bond. So, inactive storage form of proteins are that is called glycogen. They are masked by the small peptide, and they are secreted by into the duct, because you see that there is a salivary gland here. There are two parotid glands, so submaxillary submandibular gland. In the pancreas also there is pancreatic acinar cells. So, these secretions are secreted into the duct. Perhaps you have noted when you have taken a sour, material, maybe lemon juice, you feel something on both the sides, and there is juice coming out between the two sides of between the cheek and the tooth. That salivary gland duct opens into that area. So, they are secreted by exocytosis, and they are activated by proteolysis, means that particular masked oligopeptide will be removed. So, they are that is called proteolytic activation, and in case of pepsinogen, 44 amino acids are removed from the original pepsinogen molecule. So, that removal of the 44 amino acid converts it into a active enzyme molecule, that is pepsin. Similarly, for trypsinogen, there is a hexapeptide which marks the site. So, that is called a trypsin. Chymotrypsin is a bit tricky, because you see chymotrypsin, if you look at the thing, the whole chymotrypsin molecule are like this, and there are sites, the yellow colored sites are disulfide bonds, like insulin. Even after breaking down, there is disulfide bonds link that peptide two chains of insulin. Here, what is happening? There are cleavage sites. So, finally, actually, these sites are clipped off. So, this chymotrypsinogen is in this form. So, there is a site called, it is clipped off, that is called trypsinogen, and then there is further cleaving of this side. So, these two red colored materials are actually removed. So, ultimately you get 3 fragments. So, these 3 fragments are joined together, and they form that active, that is alpha chymotrypsin, and that alpha chymotrypsin is the active chymotrypsin. So, from chymotrypsinogen, by a very complex procedure, 3 fragments are produced, and then the 3 fragments remain together, connected to each other through a disulfide bond, and they are approximated, and then the active site is actually opened. So, everything is not so easy. So, from zymogen to active form, pepsinogen is activated to pepsin by gastric hydrochloric acid, and once pepsin is formed, pepsin itself acts as a proteolytic enzyme and causes its own activation. So, first stage, pepsinogen to pepsin is formed by the gastric HCl, and then the pepsin itself causes its own activation. Similarly, in the case of trypsinogen, So, trypsinogen basically is produced by the pancreas, trypsinogen, chymotrypsinogen, elastase and procarboxypeptidase. These are the 4 enzymes which are produced by the pancreas. 3 organs are totally involved in protein digestion, as I told you: stomach, pancreas and intestine. So, these trypsinogen, chymotrypsinogen, procarboxypeptidase, proelastase, they are formed by the pancreas. So, after it comes out through the pancreatic duct into the intestine, trypsinogen is activated by the enterokinase or enteropeptidase enzyme, which is present on the mucosal cell, this side. So, enterokinase activates trypsinogen into trypsin. Now, once trypsin is formed, trypsin basically catalyzes the conversion of trypsinogen to chymotrypsinogen, procarboxypeptidase or proaminopeptidase to carboxypeptidase or aminopeptidase, proelastase to elastase. So, first conversion important part is trypsinogen activation. So, trypsinogen activates all other 3 enzymes, and proteases are the brass border proteases of the intestinal mucosa are enterokinase and aminopeptidase. Remember, carboxypeptidase or procarboxypeptidase is formed by the pancreatic juice, but aminopeptidase are formed in the intestine. This is the difference. So, this is one part you must remember. Carboxypeptidase is formed by the stomach, by the pancreas, but aminopeptidase by the intestine. So, in the lumen you see that trypsinogen is coming, the enterokinase is present in the mucosal cell. All these 3 enzymes, say trypsinogen, chymotrypsinogen and procarboxypeptidase is coming into the small intestinal lumen. In the second stage, trypsinogen is getting activated into trypsin. Now, this same trypsin is acting on chymotrypsin, dinogen to chymotrypsin, and then procarboxypeptidase to carboxypeptidase, and they remain the active form. Okay. So, now you have to remember this part, that which bond is cleaved by which enzyme, that is the heart of that of this today's class. Now, there are books which I have seen some books. Harper 32 edition is saying something. Your traditional DM Vasudevan's books is saying something, but somehow pepsin is having a broad range of specificity. It cleaves the peptide bond of aromatic amino acids and the branch chain amino acid. It is not written very clearly in Harper books, but I searched in detail. There is a highest cleavage probability site. So, when we talk about cleavage of the bond, peptide bond, deliberately I have written this thing, that P1, P2 side, just to orient you with the term. I could have written carboxyl side, that is what is written in the book on the carboxyl side of the amino acid, which is easy to understand, but to make you aware that P1, P2, P dash site, suppose this is the site, enzyme site, this is to be cleaved, okay. So, that is basically a sessile bond. So, the structure which is present at this just on the N-terminal side is called P1. Next amino acid is called P2. And just C-terminal to that sessile bond, which is cut, which is to be cut, is called P1 dash, P2 dash, P3 dash, like this. So, this is called a sessile bond, which is cut. Is it? Now, there is a highest probability if you can see that phenylalanine, leucine and methionine if it is at the P1 site, and the phenylalanine, tyrosine and tryptophan at the P1 dash site, then there is a high probability of cleaving that bond. So, you understand what is called a sessile bond. That sessile bond is basically the peptide bond which is to be cleaved, that is this one. This one is to be cut, that is P1, P2, P1 dash, P2, P3, like this, okay, and this is called so you have to remember that thing. So trypsin basically cleaves the carboxyl group of basic amino acids. This is written in the book, you need not write down, only just to emphasize about the P1, P2 and sessile bond I wrote it. Pepsin basically aromatic amino acids and your branch chain amino acid. In addition, there is a methionine also. So trypsin basically cleaves the basic amino acids, chymotrypsin aromatic amino acids at the carboxyl side, and elastase small neutral amino acids like glycine, valine, and then your serine, leucine, isoleucine. Exopeptidases, as I have told you, it cleaves the terminal part of the polypeptide bond. So at the end you will get different things. One is tripeptide, dipeptide, aminopeptidases is this one. And there are two types of aminopeptidase. One is leucine aminopeptidase, which cleaves preferentially when the terminal amino acid is the leucine. But it can also cut, as I told you, that we talk that enzyme is very specific, which is not true in all cases. See, it preferentially cleaves the leucine here, but they can cleave the terminal part of other amino acids as well. Similarly, proline aminopeptide is there, that removes the proline. Carboxypeptidase are produced by the pancreas. There are two types of carboxypeptidase. One is carboxypeptidase A. Remember, A is for aliphatic and aromatic. You can note down. Carboxypeptidase B is for basic amino acids, but it can also cleave that neutral amino acid also. So everything cannot be remembered so easily. So whatever I thought that it is better to memorize that carboxypeptidase A is A for aromatic and aliphatic, B for basic amino acid. So you see here dipeptidyl peptidase. On the amino terminal, if this bond is cleaved, that is cleaved by the dipeptidyl peptidase. And if on the carboxyl side, if this dipeptide is cut, that is called by peptidyl dipeptide, and there is a practical application that DPP4, this is called DPP, dipeptidyl peptidase. So DPP is basically a digested product of dipeptide, and DPP4 is one enzyme that activates GLP-1 and GIP, which will be taught during diabetes class, which basically lowers blood sugar, blood glucose level. So if we use a DPP inhibitor, that will cause further lowering of the blood sugar, and DPP inhibitors are used as drugs. This is where I am linking biochemistry with pharmacology. So DPP inhibitors are gliptins, basically they are used in the diabetic patient treatment. Similarly, peptidyl dipeptidase, basically it is an octapeptide which is made into decapeptide. So there is 2 peptide lower down which increases blood pressure. So DPP inhibitors, the peptidase is basically AC inhibitors. They are used for lowering of blood pressure, hypertensive patients. These drugs are used. So mouth, there is no digestion of the protein, as I told you. So protein digestion starts from the stomach, and stomach acts as a hopper. It is a temporary storage site, and there is further grinding, and then the foods are propelled into the intestine, that is duodenum. So there is a regulation of stomach secretion by the hormones. Three key hormones are involved. One is gastrin, histamine, and acetylcholine. These all things will be taught during our physics. And if you need one more class, I can take it also. So basically, hydrogen ion is pumped. That hydrogen ion is called pumped into the stomach cell. That's why it is called a proton pump. And from the stomach, that is also glycoprotein intrinsic factor is formed. So it acts as a hopper or temporary storage. Hopper means something like a funnel-like structure. So it slowly delivers the food content into that duodenum. So gastric juice pH is around 0.9 to 1.5. So pepsin acts during that pH. So in the stomach, there are 3 types of cell. One is parietal cell, which secretes HCl. Chief cells are produced. Note down, chief cells of the stomach produces pepsinogen, and the columnar cell secretes mucin. So stomach is producing gastric hydrochloric acid. So that hydrochloric acid is not damaging the stomach wall. Pepsinogen is also activated there, so it can also digest. It is unable to do that thing because it is protected, because columnar cell produces mucus, and that mucus acts as a protective barrier of the mucosal lining. So normally acid peptic digestion doesn't occur in normal individual, but if you indulge in producing, or if the disease produces excess amount of hydrochloric acid, obviously the mucosal barrier, protective mucosal barrier will be destroyed, and that will cause an ulcer formation. So there is gastric juice. This will be taught in the anatomy class. So opening into the cell, this gastric lumen. So pepsinogen is produced, available in the gastric lumen. Now pepsinogen is getting activated. You can see there is a control secretion. Now pepsin is basically an aspartic protease. Aspartic protease means at the active catalytic site aspartic acid is there. That is called, why it is called aspartyl protease or aspartic acid protease. So it breaks down the formation. So renin, much talked about that it is present in the infants. It curdles milk. It produces paracasein to calcium paracasinate, which is insoluble form. It allows the protein to remain in the stomach for a longer time for digestion for pepsin to act. But remember, it was produced in the animal's fourth stomach. Much talked about material, but not in all human beings. I tried to look at the literature. Actually, there is a 1967 literature which says that there is a fourth stomach of the cup where it is present, and there is a human study on in 1987 where they have taken 5 premature 10 days infants, and they have found that chymosin is another name for renin. It is present only in 2. Almost exclusively pepsin, like an adult, is found in 1, and mixture of both chymosin and pepsin is found in 2. So renin is a controversial issue. I must say that at this stage you must not know all these things, but to give an insight that whatever is taught today will not be taught tomorrow. Things will be changed. So you remember at this stage, Rearing causes, curdling of milk, paracaseinate formation. It allows pepsin to give more time to act. So, this is the first part of the duodenum. You see the duodenal mucosa is looking like this. Now, when that mucosal barrier, that protective mucosal barrier will be destroyed by any means due to excess HCl production or due to drugs also. So, this type of ulcers will be formed. So, if you put an endoscope, you will see an ulcer like this. So the ulcer looks like this, which is not the normal cell. Normal structure is like this. This is the normal structure of the gastric mucosa. Now, if that is broken down, so that is called this parietal cell is under control of acetylcholine, histamine. This I wanted to proton pump that hydrogen ion is pumped through that proton forms basically hydrochloric acid. So, proton pump inhibitors, most of you have taken omeprazole, pantoprazole, they basically stops the proton pump. They act as a proton pump inhibitor and they decrease the gastric hydrochloric acid secretion. So, protein digestion in the stomach, small intestine. So, I told you all these things about all these things. So, pancreatic juice is basically these 4 enzymes which are active on the at the alkaline pH. Intestinal juice is also at the alkaline pH. So, when there is a digestive disorder like digestion, they cannot digest the food properly. So, these digestive enzymes are given as medicines. As some of you have perhaps taken that enzymes for digestion also. So, absorption in the intestine is basically by the sodium dependent mechanism that requires an ATP along with the amino acid. Sodium is also pumped out, the sodium ATP pump is active. So, sodium independent carrier transport mechanism is there. One is for neutral, one is for acidic, one is for basic, and fourth one is for amino acid, and fifth one is beta alanine. Beta alanine is present in the carnosine, which is present in the meat. So, beta alanine is produced by the L-histidine and beta alanine. So, in the intestine, there are a large number of amino acids which will be available for absorption by the mucosal cells. So, there will be a competition among all the amino acids, which one will get absorbed faster at a most rapid stage. So, at the end dipeptides and tripeptides are formed. So, usually the amino acids are absorbed by the intestinal mucosal villi cells. At times dipeptides and tripeptides are also formed, absorbed, and these dipeptides and tripeptides are again broken down within the intestinal cell into amino acids and they are finally absorbed. At times small fragments like of protein that is polypeptide, not polypeptide, short fragments of peptide are absorbed through the cells or paracellularly. Paracellularly means between the two cells. So, these enter into the circulation and then they cause allergic reaction that causes food allergy. So, Mr. cycle is one thing which is not very much important, but can come as a short note because amino acid is absorbed through glutathione. This enzyme, tripeptidyl gamma glutamyl transferase enzyme is present on the membrane and this requires glutathione for amino acid transport. Glutamyl amino acid is formed and finally amino acid is released here. Now, to allow absorption of one amino acid, 3 ATPs are required. So, it is an energy consuming procedure. So, at the end glutathione is regenerated for the cycle to operate. So, that is called a Mr. cycle, Master cycle, whatever you say, but there is an energy dependent mechanism. You read this thing in the book, from the book, there may be a short note on this Mr. cycle, Master cycle also, Master cycle. So, dipeptides and tripeptides are formed, they are hydrolyzed by the intestinal cell I have told oft repeatedly, and some proteins are, peptides are absorbed so paracellularly, and then they cause food allergy. Normally, protein food is called fish, meat, they can cause food allergy. So, there are these relatively large peptides that are produced paracellularly or transcellularly, and when you will be taught about the immunology, the protein size will be something, a large size of protein will be required to produce an allergic manifestation that will act as an antigen. So, that can cause an immune response causing allergy. So, summary, that protein, as I told you, from stomach, intestine, that part I have discussed in details. Now, clinical conditions which is associated with basically digestion and absorption. So, any disease which is they are in the stomach, intestine, pancreas, that will cause digestion and absorption problem. So, what are the conditions? Either allergic to certain proteins which is common, milk or meat, disease of the digestive tract. It can be any disease which will impair. So, yes, ek minute lagbe. So, like cancer, like cystic fibrosis, or if the organ is removed due to any disease. Suppose there is a cancer in the stomach. So, the cancer in the small intestine, either partial, subtotal or total removal of that particular organ, from the body will impair definitely digestion and absorption, because that part is absent. So, there will be a serious impairment, malabsorption syndrome. It cannot, the digested food cannot be absorbed because of diseases of the intestinal tract. And in your book you will see that there are these 4 diseases which are written, but they are not that common. One is heart nerve disease, that is tryptophan absorption disorder, and immunoglycinuria, there is a defective absorption. Glycinuria means this is in the urine, but that means it is related to kidney, but the same defect is there in the intestine also. So, immunoglycinuria, amino acids are basically proline and hydroxyproline and glycine. So, there will be a defect in the absorption of proline, hydroxyproline or glycine, and cystinuria, not cystine, S-C-Y-S-T-I-E-N-E. Cystine is a single amino acid, cystine is a dimer of cystine. So, cystinuria is there, and 5-oxyprolinase deficiency, it is present in the Mr. cycle, and these are all the rare things. The common things are like this, diseases of the digestive tract, and the whole DM course is basically, DM course means after MD, there is a DM course for 3 years which are dedicated to handle all those diseases. So, it cannot be covered in just 1 minute time. Okay. Thank you very much. This is a protein rich food, I will say that it is very much lucrative. So, do not take this type of food, because carbohydrate requirement is 50 to 70%. Protein requirement is 1 kilogram per kg body weight, that is the protein requirement. If you take excess amount of protein, the protein will be simply broken down into urea and it will be excreted to the urine. So, excess protein intake more than the calorie intake will be an useless food. It will be, you can take it for your own satiety, but it will not serve any useful purpose except urea formation and excretion through the kidney. Okay. Thank you.